Hi, everyone. My name is Daniil Gataulin. I'm one of the Senior Research Analysts covering biotech here at Chardan, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Nadia Waheed from Beacon Therapeutics to the second series of our virtual ophthalmology summit, where the focus is on retinitis pigmentosa and other inherited retinal diseases. Nadia, welcome.
Thank you so much, Anil.
Yeah, so to start things off, could you please provide a brief overview of Beacon Therapeutics and the ongoing programs?
Absolutely. So Beacon Therapeutics was just recently formed this year. It's Syncona supported company. Our goal is to provide gene therapy based treatments for broad as well as rare retinal diseases. And our lead program was acquired from AGTC. It's AGTC 501, which is a gene therapy targeting X-linked retinitis pigmentosa. And this has, we have results coming from a phase 2 clinical trial right now. It is our most advanced clinical program. In addition to this, we have a couple of preclinical programs that we have, made public. One of those targets broad disease, dry age-related macular degeneration. And then we have another preclinical program that's been in-licensed from the University of Oxford that looks at a specific cone rod degeneration, inherited cone rod degeneration. And then outside of that, we have a pipeline that generates target-based approaches, which is also a gene therapy pipeline that does target-based protein approaches that we also have as part of our pipeline.
Got it. Thank you. That was very helpful overview. So I'd like to spend majority of our time talking about your XLRP program, your related indication. First of all, can you talk about that indication? And in your opinion, what fraction of the RP market do you expect to benefit from AGTC 501?
Absolutely. So, as you're aware, AGTC 501 targets X-linked retinitis pigmentosa. And X-linked retinitis pigmentosa, it's a unique disease. It occurs primarily in young men, although there's more and more evidence now that, some women may also, who have the gene may also show some signs of it, but primarily affecting young men. And these young men progressively develop more and more difficulty with their peripheral vision and night vision starting earlier on in life. In their teens, usually they will start noticing this. And then it progressively constricts their visual field. The visual field gets smaller and smaller and smaller until, they can lose central vision. And many of these people go into legal blindness in their 40s. So it really is quite a devastating disease. It's about 10 to 15% are the quoted numbers of patients with retinitis pigmentosa have X-linked retinitis pigmentosa. But the very significant piece about X-linked retinitis pigmentosa is that unlike, say, autosomal dominant or autosomal recessive RP, this does tend to be a relatively severe form of retinitis pigmentosa and is the one that, starts impacting young men in the peak of their productive lives and then just gets progressively worse. It's just really, really devastating from in terms of who it affects and how severely it impacts these patients.
Got it. Understand. And speaking of AGTC 501, can you provide an overview of that therapy and what do you believe differentiates it from other programs in development?
No, absolutely. So AGTC 501, was a program that, as I mentioned, was licensed from AGTC, which was a Boston, Massachusetts-based biotech company. They had developed this gene therapy program. It's basically a modified AAV2 based gene therapy that is delivered subretinally in using a surgical approach in the eyes of patients with X-linked retinitis pigmentosa. Some of the differentiating factors are just the fact that the AAV2 used over here allows for very high transduction levels. in cells, in photoreceptor cells specifically, which are the ones that are targeted. It is, it's a pan photoreceptor cell expression, so both rods and cones. And the reason that this is important is because while it's a rod cone dystrophy, while the disease starts in rods in the later stages, as it progresses more and more centrally, more and more cones become involved as well, and that's how patients end up losing their central vision. But this gene therapy targets both the rods and cones. We have taken the time, and AGTC, as it developed this, took the time to develop a full-length protein. So the DNA, unfortunately, for full-length proteins generally is not very stable. And so a lot of work was put into stabilizing that DNA for the full-length protein, so that the protein that's produced is the full-length native protein that normal cells produce. It's not a truncated version as some of the other ones are. And the advantage of the full-length protein is that it tends to work really well in both rods and cones. And so, we believe that it will target both the rod component of this rod cone dystrophy as well as the cone component of the rod cone dystrophy. So that's the scientific piece. And then the AGTC 501 has gone through several studies. The Horizon study, for example, was the phase 1 dose, first in human dose escalation clinical study that really helped identify the maximal tolerated dose, as well as the dose that was taken forward into the phase 2 studies. Now, the phase 2 Skyline study tested 2 doses, and the 3-month data that has been presented so far found that the higher of these 2 doses appear to be very effective in terms of preserving retinal sensitivity in these patients as measured in microperimetry. There were also strong signals, although not statistically significant, when we looked at the maze test, or the visual navigation challenge test, where these treated eyes were doing significantly better, or were doing much better than untreated eyes in terms of being able to navigate through a visual navigation challenge such as the maze. So overall, robust and convincing data showing that this is working in these patients, and of course, future trials planned to show this. Of course, one of the pieces that these clinical trials has really helped us understand also is what is the cohort of patients in which this gene therapy trial is most likely to work, and what are patients that are, for example, too far advanced, and therefore perhaps this gene therapy will not work as well in that cohort of patients, or maybe perhaps they're too far along to rescue. So there's a wealth of data that we have coming out from the Horizon and the Skyline studies that has enabled us to understand, what the ideal patient population is like. And of course, one of the nice things that's been presented recently is the 24-month Horizon data that shows that there is a persistence of effect, that, really you start seeing an effect at 3 months, that effect persists out to 12 months. And now we have good data showing that actually it persists out all the way out to 24 months. We're still seeing that durability of effect, which really is a promise of gene therapy, right? Is that one-time administration, long-term durability of effect.
Got it. Thank you. So I wanted to go back to you and talk just a little bit more about the construct itself. So you mentioned that you're utilizing a full-length RPGR protein or RPGR construct that expresses the full-length protein. I understand that it's always better to have a full length versus truncated, but to be more concrete, can you perhaps summarize some of the preclinical data to highlight the actual benefits, the actual scientific benefits that are achieved with the full length protein?
Absolutely. So obviously the biggest advantage of the full-length protein is that it is the natural protein that's produced inside the cells, and unlike so typically I think the preclinical data has demonstrated that truncated proteins are actually really good at causing functional production and causing functional rescue in rods, but perhaps not nearly as good in cones. And this has to do with the ORF10 repeat region that is really important in cones, and that is truncated out of the truncated construct. Now, the full length, of course, has this region. Therefore, the preclinical data out of animals shows that that it is equally efficacious in rods as well as in cones. And so we think that this should be able to target the whole spectrum of cells that are involved in this rod cone dystrophy, and especially should be able to prevent people from losing visual acuity in the later stages of the disease, and in maintaining their visual acuity in the later stages of the disease. So, the theoretical piece as well as the preclinical data seems to show very robust rescue in both rods and cones with the full length protein, unlike with the truncated protein, where the effect is much stronger in the rods than it is in the cones.
Got it. Understand. And in terms of the administration, so you mentioned that it's a subretinal, and does this therapy require the prophylactic use of steroid regimens to reduce the risk of inflammation?
Yes. So, as you know, subretinal gene therapy is associated, with low levels of inflammation, generally much lower levels of inflammation than you would get from, say, intravitreal gene therapy or indeed systemic gene therapy, because it is in an immune privileged space. However, having said that, there is still, as studies have shown with administration of AAV gene therapy, there is a risk of inflammation. And so patients who were treated in both the Horizon and the Skyline study did get systemic steroids with administration of the gene therapy. And these systemic steroids were then relatively rapidly tapered off, but they did get prophylactic systemic steroids to prevent inflammation from happening in the eyes of these patients. Now, the safety profile in terms of inflammation of both the Horizon and the Skyline studies are excellent. They're very good with really no evidence of severe inflammation in the eyes of these patients noted. However, future studies will continue to use a prophylactic steroid regimen. What we are doing is we're also in future plan studies exploring whether we can go down on the quantity of systemic steroids being given to these patients, but that is obviously still an open question. And for now, until we get the results of those open label studies that really interrogate the safety of using less systemic steroid prophylaxis, we do anticipate that patients will need systemic steroid prophylaxis, albeit for a short amount of time post-administration, to prevent inflammation from happening inside the eye.
Got it. And one more question about the contract, the therapy itself. So 501 utilizes AAV2 vector. Wanted to ask, what are the pros and cons of using this vector for XLRP indication?
Absolutely. So I think one of the big pros of AAV2 is that it is a predicated vector that we really understand very well. It's clinically predicated, it's regulatorily predicated with the Luxturna approval. We have decades of data on AAV2. We know that it is safe, it is well tolerated. We know that it does result in long-lasting expression and in long-lasting production of protein. And from what we've seen from the Luxturna data, from what we've seen coming out from the Biogen data, for example, as well on their X-linked retinitis pigmentosa program. So I think that those are the advantages of an AAV2 construct. Now, some of the challenges with the AAV2 construct are that it does not generally result in as robust transduction as, say, an AAV8 would, right? However, keep in mind that this is a modified AAV2 vector, and the the preclinical data that we have from this modified AAV2 vector does show that it actually causes transduction better than even an AAV8 construct. So, and that really is the magic sauce of what was available at AGTC, and what we're using going forward in Beacon is that this is a modified vector that results in very robust transduction and production of proteins in the photoreceptor cells. And I think that's the magic of using an AAV2, but then using this modified, customized AAV2 that results in this very strong transduction and high levels of protein expression.
Got it. Understand. All right. I'd like to switch over and talk a little bit more in depth about the clinical data. So, as you summarized, the phase 1/2 Horizon trial results have been presented. The phase 2 Skyline trial is ongoing, and I believe the company is expected to publish or present data, 12 months data, in the second half of this year. So I wanted to ask if you could guide us in what we should be focusing on ahead of that presentation.
Absolutely, yes. So the Skyline 3-month data has been presented, and the 12-month data is expected to come out towards the end of this year and be presented depending on meeting scheduling, either towards the end of this year or beginning of next year. So the 3-month data showed very robust showed a very robust effect in the high dose group of our patients when we looked at their microperimetry response in terms of visual sensitivity. And this is compared to untreated control eyes. Or untreated fellow eyes, I should say, because there was no true control group in the Skyline cohort, but there was untreated fellow eyes that we were comparing the microperimetry response to. It also showed a very nice dose response. So in the high dose, treated eyes were compared to the eyes treated with the lower dose. There was a statistically significant difference in terms of response and visual sensitivity. And so I think for us, the big thing to look out for will be whether this response is sustained at the 12-month time point, which is the next cohort of data that's going to be coming out. Now, based on the Horizon data, when we look at the parallel doses in the Horizon study, which is dose groups 4, 3, 4, and 5 in the Horizon, or 4, 5, and 6 in the Horizon study, which are the doses that are that were brought forward. 2 of those doses were brought forward into the Skyline study. We did see the effect persisting out to 12 months and furthermore to 24 months. So if we're mirroring that same data in the Skyline study, that is what we would expect, but time will tell as that data becomes unmasked and as we analyze that data. So that is one piece that I would look for, is to see if that visual sensitivity response persists out to the 12-month time point. Additionally, like I said, trends in improvement of the visual navigation maze. So that is another piece that I would like to see in the 12-month data. And then I think the third piece that we need to focus on is what the safety profile of the medication looks like. And that's also a piece that I think will be really important to look at as we look at the 12-month data, is whether the clean safety profile that we have at 3 months is replicated out to the 12 months time point. And keep in mind, of course, that as far as safety goes, a lot of the inflammatory side effects, which are the biggest risk with these patients, happen in the first, in the post-surgical period. So, generally, if data looks clean at 3 months, it's likely to look clean at the 12 months time point as well. But of course, time will tell as we look at those 12 months time points what that data looks like.
Got it. And can you comment on any additional metrics that are included in the Skyline trial that were not a part of Horizon?
So the Skyline trial really was to look at efficacy, versus the Horizon trial really was designed to look at safety. So we have many more patients in these dose groups in the Skyline trial, right? So I think that's one of the pieces that we will have much more robust data and a much larger number of patients. When we look at the Skyline data. Because it was designed to look at efficacy, there was a large group of endpoints that we looked at, and that will also be, coming out. So, for example, FST, right? Full field sensitivity threshold testing is another one that we're excited to look at and see, what that data shows, because that data, as you know, shows a summated functional response of the entirety of the retina. And to be able to, evaluate that data at the 12-month time point, I think will give us a picture of, what this gene therapy is doing. Right. And then other parameters that I'm really excited to look at are looking at the OCT parameters and looking at, for example, the ellipsoid zone in these patients, really evaluating the microstructure of the photoreceptors. And we have some very preliminary data from the Horizon study that's actually getting presented at the Retinal Degeneration Summit later on this year. But really looking at that in greater numbers and in greater detail, I think in the Skyline study will be something very, very interesting and unique to look at. And of course, we're also looking at other functional parameters, such as looking at the hill of vision, for example, and seeing how that changes over time and how that holds out over the course of a year, and what kind of efficacy signals we see in there. So really a whole plethora of efficacy parameters that are being looked at, both functional as well as structural parameters that we're looking at in this Skyline data.
Okay, got it. Understand. And so there've been quite a few patients who've been dosed with this already. So wanted to ask if you could share any patient or physician feedback that we received to date.
Yes, we are very involved with patient organizations and have had patients and patients' parents come and speak both internally to our group, as well as had patients and go out and speak at conferences, at meetings, at patient advocacy groups. I think patients are really enthusiastic about this therapy. I think that patients, especially who have received the treatment, are noticing the impact that it has on their lives, and are advocating very strongly for this. I think patients also understand the potential advantage to having that full length protein construct that reconstitutes a natural protein within their cells, and have been really interested in seeing this go forward. So far, the patient feedback that we have gotten has been generally very, very positive in terms of wanting to see this progress and wanting to see this move forward.
Excellent, got it. So in terms of the next steps for the program, I believe the phase 3 trial, the VISTA trial, is being planned currently. Wanted to ask if there are any detail that you could share, maybe potential details about the study design, and when do you actually expect the enrollment to begin?
Great question, Daniil. So the VISTA study, which is a phase 2/3 study, it is meant to test 2 doses of the gene therapy product against a cohort of untreated controls. So this will have a true control group. So what we do want this to be is a very well-controlled clinical trial in which we test 2 doses of the gene therapy product against untreated controls, and in which patients are randomized to receiving either a dose of the gene therapy or untreated control, or an untreated control group. We do plan on going with the dose that we know is already efficacious in the Skyline study, and another dose that will be slightly lower than that higher dose that we know has good efficacy, kind of to try to elicit and winnow out more of a dose response in these patients, and try to figure out what the minimum efficacious dose is going to be for these patients. And I think, again, we'll go in with a broad cohort of endpoints. I think the details of the study are still being ironed out as we're in conversations with the regulators. But, again, follow patients up for a year as a primary endpoint, and then keep generating data all the way up until probably 5 years or so, just to generate both the efficacy data, but also, very importantly, with gene therapy products, to generate the safety data through this long-term follow-up.
Okay, got it. And in terms of the regulatory plans, I guess, do you think the results from VISTA trial, together with the results from the Horizon and Skylight, will provide sufficient data for regulatory submission?
I think we are in conversations with regulators right now to understand, what all, is needed for, what all they would like to see us present to them. I think there's certainly broad agreement overall with the European regulators around, what they would like to see in terms of an efficacy signal and what endpoints they would want to see. And again, like I said, we are continuing these conversations, and we hope to have more information towards the beginning of next year as we roll out VISTA around some of these, parameters that we hope to be able to interrogate.
Okay, I understand. In terms of the company's manufacturing capabilities, can you talk about whether you're manufacturing 501 internally or if you're working with the external development organizations?
We have worked with external development organizations to produce some of the material that has been used in the clinical trials so far. I'm happy to say, though, that at this point we have in-house manufacturing capabilities that are able to take care of developing the commercial product for us. And so, most of the manufacturing going on forward will be almost entirely in-house.
Got it. Excellent. And I'd like to conclude our session with a couple of more general questions, if I may. So first, as a recent, spin out, wanted to ask, what do you expect the current capital to carry you through? Which milestones?
So I think that that is not information that has been publicly released. However, I will say that we have, enough cash in hand to be able to kind of continue our program, and are just, really excited, as well as, our investors are also really excited about the potential that this recently acquired program, the AGTC-501 program, brings to Beacon Therapeutics. It is definitely our lead program, so we're all very strongly invested in it and leaning strongly into it, and really believe in this, and believe that it will, produce the kind of results that we hope to produce for patients who have this devastating and blind disease.
Got it. I understand. And the last question that I like to conclude this session with, what do you think investors are still potentially missing about Beacon Therapeutics?
So I think one of the most interesting pieces around Beacon Therapeutics is kind of the depth of experience that the team brings. Both we have very experienced investors who have played and worked in this field before. The management team for Beacon Therapeutics is incredibly experienced. I'm Dave Fellows, who is the CEO, has, a long history in big pharma, was also, history with Nightstar. So, history in this very area before. So we really do have amongst us a group of very strong veterans in this field who together bring, decades of experience in gene therapy, both in gene therapy for rare diseases as well as in gene therapy for broad diseases. I think we have a very exciting portfolio that targets lots of different disease entities. And so just, I think that you can't discount the fact that, when you have a strong leadership team, and then you have, a strong pipeline and a broadly diversified portfolio, all the way from stuff that's, in clinical trials to go into market soon, to stuff that's being investigated preclinically, it creates a very strong company.
Got it. Understand. Excellent. Thank you very much. I believe that brings us to the end of our session. Really appreciate your time, Nadia, and look forward to catching up with you again. Thank you very much.
Thank you so